Regulatory motifs are sequences of DNA that are found in the genome of every organism. So just like in humans, these regulatory motifs are also found in other model organisms and being worked on to find the functions in model organisms. So model organisms are being used to find the functions of these regulatory motifs. So these regulatory motifs are generally, they are present in the promoter region and they are helpful in the expression of the genes. So these motifs, they facilitate for the binding of the transcription factors, and these transcription factors recognize these specially sequence of DNA and bind on them, and in turn they help in the binding of the RNA polymerase for transcription process. So already we know that these regulatory motifs are very important, and for example in this case we will discuss the Drosophila fruit fly, and we know that in Drosophila fruit fly, just like in humans, there are many immunity genes that are present in Drosophila. So in Drosophila there is a specific sequence, this one TTC, GGG and so on. So these sequences are regulatory sequences and they help in the activation of immunity genes in Drosophila. These sequences are basically the NFKB binding sites, and this NFKB is abbreviation of nuclear factor kappa light chain enhancer of activated B cells. So these sequences are very important for the activation of immunity genes in Drosophila. And as I have told you that these are the very important regulatory motifs, and generally these regulatory motifs are being recognized by transcription factors. And when transcription factors bind to regulatory motifs, they in turn help in the binding of RNA polymerase. So scientists can perform many experiments to find the regulatory motifs like this. So for this purpose they can perform a lot of genomic studies in which they can find the specific sequences that are repeated many times. So the sequences that are repeated many times, they have some message in them, just like this NFKB specific binding sites. So after the genomic studies, scientists can find sites like these, and they can also determine the location of these sites. But in our case, we suppose that we do not know either the location or the sequence of these NFKB sites, and we have to find this by using some method or algorithm, and we have to use certain computational approaches. To find more about the regulatory motifs, how we can decode the information or find the information of some specific sequences that are repeated time and again in the genome of any organism, we should know about this gold bug theory that is written by Edgar Allen, and it provides some clue to find the DNA motifs in the genome of organisms. So it is the story in which one of the characters of that story find the parchment written below. So we can see it's a very strange alphabets, numbers written here, and we could not recognize this. So basically it is the coded information that is written in the form of these integers. So here we can see that the semicolon 48 is repeatedly used in this coded information. So one of the characters that is written in this one has coded this. He knows that the information written by the character is an English speaking person, and in English the word the is used very frequently. So he assumed that semicolon 48 must be used as the. So by using this method, he has decoded this information and he came up with this one. So after decoding this information to the somehow, he has explained the position of semicolon 4 and 8. Here we can see that he has neither he used this semicolon 4 8 for the, but wherever there is a semicolon, either it is with 4 or 8, he used the letter T for semicolon. And similarly, he used the digit 4 for H. Wherever there is a letter 4, he used the alphabet H for 4. And similarly for 8, he used the alphabet E. So in this way, we can also use this clue to find the regulatory motifs in the DNA sequences. So we can see whether in the DNA sequences, some sequences might be repeating several times, and the repeating sequences must have some information, and this information is converted into a signal, and that signal is used for the binding of specialized transcription factors. In this way, the gene is activated.